Sejam bem-vindos ao Fibrenamics Impulse at Home. Esta sessão está a ser transmitida igualmente para o Facebook, mas para poder partilhar as suas opiniões e colocar questões aos oradores, deverá registrar-se no evento em fibrenamics.com e acompanhá-lo a partir da plataforma Click Meeting. As perguntas mais relevantes serão respondidas pelos oradores após cada intervenção. Esta sessão é especial. O nosso orador convidado não domina a língua portuguesa e, por isso, irá falar-nos em inglês. So, we welcome Dmitri Petrovykh, Corporate Expert at INL, International Iberian Nanotechnology Laboratory.
Hello, good morning. I thank the organizers for the opportunity to tell you this morning a little bit about how technology and nanotechnology in particular is being used in the response to the COVID pandemic. So as part of my job as a corporate expert at INL, I have to promote interdisciplinary collaborations both inside INL and of INL with external partners. And of course, intrinsically, most of the activities related to the COVID pandemic response, they have to be interdisciplinary. So it is, I'm very glad that I have a chance to talk to an audience with different types of expertise today. So before I go with the presentation, just a quick introduction of my home institution for those of you who are not very familiar with it. So I come from the International Iberian Nanotechnology Laboratory. Our Director General is Professor Lars Montelius, and we have been co-founded and are currently partially funded by the governments of both Portugal and Spain. Which means that the international part of our name is not just the word, but actually it's our legal status. So we are an international organization with headquarters in Portugal. We're still a relatively young organization, but effectively we have been operating for about 10 years. And at this point, we have about 400 researchers from more than 30 countries. But of course, again, right now, many of us are working from home, although some of the laboratory activities have resumed a couple of days ago. So we're trying to get back to work. In terms of our ability to respond to things like events like the COVID pandemic, of course, what helps is that INL intrinsically is a very highly integrated research organization. And it's both at the internal level, where we basically have life scientists working together with physical scientists and taking advantage of the micro and nanofabrication facilities that we have. And of course, we have very strong engineering groups and system integration groups that put everything together. And of course, we also, because of the high level of internal integration, we are quite good at integrating not only our own knowledge, but also working together with partners. So the examples the images that you see at the bottom of the slide, they sort of give you an idea that our work really spans the full range from basically the fundamental questions about, for example, biointerfaces and molecules at biointerfaces, which I'll mention later in the talk, or for example, questions of how nanoparticles might interact with cells, to using nanotechnology as the basis for proof of concept devices, and then to making the fully integrated platforms that are portable and actually can be tested in the field. So after this brief outline, after this brief introduction, basically I will cover 3 topics in my presentation. So one of them, I will start by introducing the technology that is currently used in response for the COVID pandemic, and particularly the diagnostic technology. Then I will explain how nanotechnology is already making an impact and what might be the future possibilities for nanotechnology use. And then, of course, I will finish with some examples and some ideas that INL has specifically in response to this pandemic. So, to start, what is the current technology that is used in the diagnostics? And before we talk about the technology for diagnostics specifically, I should point out, why I have chosen diagnostics as the primary topic here, as the primary area. And essentially, there are two reasons. One of them is that, of course, in principle, technology involves different areas of response to the pandemic. And that, in addition to diagnostics, that includes things like therapeutics and vaccines and the epidemiology and even the different legal and social aspects. And so, in principle, again, one can talk, probably, spend a couple of weeks talking about how technology can be used in all of these different areas. But arguably, one of the areas where the impact of the technology, both existing technology and the rapid advances, is felt most immediately is actually in the technology for diagnostics. And of course, technology will have impact also in the future, but right now, again, even since January, things have been developing very rapidly, as I will mention in a couple of examples. And another reason to focus on diagnostics is that when the World Health Organization had their forum in the middle of February, they actually identified the rapid point-of-care diagnostics as essentially the first of the 8 immediate research actions. So again, from their perspective, this is one of the areas where the research and development work and the quick deployment of the different technologies is actually paramount to ensure that we have a global response to the pandemic. Because basically, it is going to be the diagnostic technology that will allow to both understand what is happening and to treat and hopefully help recover for the patients that become infected. Okay, so I imagine that most of you have read or seen or heard something about different kinds of technology. And my apologies to those of you who have a lot of expertise in the area already, but I wanted to introduce a couple of the, let's say, explanations and introductions just to make sure that we're kind of starting from the same place. And the first sort of small comment is that in terms of terminology, it is important to remember that we have the virus, which is called SARS-CoV-2, and then we have the disease, which is the COVID-19. So sometimes people talk about the COVID virus, but that's not quite correct. But more importantly, the reason why this terminology is relevant is that when we talk about diagnostic technology, there are some diagnostic tests that will detect the virus. So they do not necessarily tell us whether a person has the disease, but they will detect the virus. Whereas there are other technologies that are looking for the signatures of the disease. Again, often any single one of them will not necessarily give us the complete picture, but we will have an indication that perhaps the disease is happening. And there will be a few examples that will try to illustrate these points. But it is important to sort of understand that at the level of diagnostics, these are, let's say, two somewhat different areas in terms of conceptual understanding. As I mentioned, the different technologies are being deployed very rapidly. So this is actually something that has been learned from the previous SARS pandemic and other, let's say, previous epidemics where we have been developing some of these technologies. So now they're actually being deployed. There is what I'm showing on the right, for example, is the head is the title of the list that Nature Biotechnology maintains at the DOI that I mentioned there. So as you see, even in the screenshot, so they actually keep regularly updating it with new information as it appears. And this is one of the many lists. So this one has some, let's say, verification of the information. So the journalists from Nature Biotechnology are updating and checking some of this information. But basically, this list has the names of the companies and the summary of the technologies that they're deploying, and also the article provides some brief explanations about the different specific technologies. But in the broadest sense, obviously the majority of the technology that is currently being used for diagnostics are the two traditional methods. One of them is Rt-Pcr, and the other one is immunoassays, and we will talk about it in the following slides. But what is interesting is that some of the, let's say, more advanced or new versions of the technology are already starting to be commercialized. So, for example, CRISPR is one of the biotechnologies that has been very popular in different areas over the last couple of years, and now it is already being deployed, as are, for example, different special variants of PCR, which I will mention as well. Depending on your background, you might also notice that right now the tools in these technological, let's say, advances primarily come from the biological side, so from biotechnology and from molecular biology. Whereas, of course, at the research level, there are concepts that are coming from many other fields, but at the moment, most of them are, not quite ready to be commercialized. Okay, so Rt-Pcr. This is probably the most widely mentioned test in the media, let's say. So this is the test that typically is done after the samples are collected with the swabs. So just as a quick explanation, so RT in Rt-Pcr means reverse transcription. And that refers to the step of having to convert the RNA sequence of the virus, because SARS-CoV-2 virus is an RNA virus, so its genetic information is encoded in RNA. And this reverse transcription step basically translates the RNA sequence into a DNA one. Let's say the practical reason for it is that we have a lot better biotechnology and molecular biology tools for handling DNA. And so it is more convenient to work with DNA for the subsequent steps. And the reason why it's called, for those of you who might be interested, the reason why it's called reverse transcription is because effectively this enzyme, reverse transcriptase, it actually performs a function that's more or less opposite to what tends to happen in the natural biological processes, where typically a sequence is copied from DNA into RNA. So that would be normal transcription. But in this case, we have to go the other way. Just as another point that if some of you were trying to do some searches, you might have noticed that in some cases, when people write Rt-Pcr, they actually refer to real-time PCR, which is something else. So this is something to keep in mind if you're trying to look for information. The second part of Rt-Pcr is PCR, obviously. This stands for polymerase chain reaction. And essentially, that technology refers to the concept that what we're doing is that we're using thermal cycling in the standard version of it to break apart double-stranded DNA into the separate strands. And then we use the polymerase enzyme to build a copy of each of these two strands, meaning that if we start from a single double-stranded DNA, after the cycle is completed, we have two copies of that double-stranded DNA. And because we basically double the number of copies at every cycle, so the process effectively exponentially increases the number. This is a method that is very effective because it is both sensitive. So again, this exponential amplification really means that even if we start with very few initial copies of the viral DNA, so even like 1,000 or 10,000 viral particles, it is possible to actually detect that they were there. And the method is also specific because in this particular amplification process actually will amplify, or let's say it is operated in a way where only the viral DNA is amplified and the other DNA in the sample is not. So from that perspective, the characteristics are very appropriate for the diagnostics. And again, this is typically the test that is administered. So when you read about testing of swab samples, this is typically the test that is done. Although in principle, in hospitals, this is also done on blood samples as well. But of course, one of the challenges is that this technique, before the pandemic, it was already in clinical use, but typically having to handle much fewer samples. So right now, there are some rapid advances that are trying to make sure that this technology becomes, let's say, faster, and or let's say that the technology can cover many more samples per day. And of course, there are several, let's say, ways that one can do it. One of them is essentially multiplexing the processing. So if you look at the plot, the vertical axis refers to the number of samples that can be done in parallel. And you can see that several of the commercial technological platforms actually allow to run multiple samples, including as many as 96 samples at the same time. So this is kind of let's say, the simplest way that technology can help to improve the throughput. Of course, by the time we go to these high multiplexing numbers, typically a high level of automation is required. So these machines typically have quite a bit of robotics to help in handling the process. And for the largest machines, in fact, the automation even of the sample preparation steps can be important, because simply feeding these thousands of samples per day into the machines also takes time and effort. On the bottom axis in the plot, you see effectively the speed of analysis, or the time to result. And again, you can see that whereas in the more conventional laboratory systems, this can be a couple of hours, but by optimizing both, let's say, in terms of technology and in terms of the biological components, people are pushing the speed of analysis to as short as about 15 minutes. So in this obviously also helps, and in particular, this can be helpful for the point of care analysis, where we would like to give the results as quickly as possible for the person being analyzed. As I mentioned already, another way that this technology is advancing very rapidly, because in principle, Rt-Pcr is a mature technology, but one of the rapid advances also comes from the fact that right now people are deploying in commercial devices some of the new variants of PCR. And in particular, the variants that are popular are the isothermal ones and so-called single tube variants. So effectively, it means that instead of having to have more complicated hardware where we have to do thermal cycling and some other separate steps, in these new variants, basically all the reagents are mixed in a single tube, and the reaction takes place at a fixed temperature and actually can happen quite quickly. So one of the popular versions is a loop-mediated isothermal amplification lamp, or more specifically, the RT lamp variant of it. And of course, again, there are many different variants that have been described in reviews and so on. And several of these variants are now being tested for introduction into commercial assays. Nevertheless, with all of the advances, actually the primary bottleneck in terms of these things being available for the clinical testing is basically the production volume. And if you look at this plot, on the bottom axis, there is the production volume for 2019 in millions of units. And you can see that actually only for a small number of platforms, the production volume is in 10 million units or more. And in fact, these two that are in the 10 million or more volume actually refer to the systems that are quite large. So they are well suited for, let's say, centralized laboratories. And these are the systems that are highly automated. They're kind of the size of a kitchen, and they require a team of people to prepare the samples. So they are quite expensive, but of course, yes, they are very efficient. And at the same time, when you look on the left-hand side, so this is where many of the newer and emerging technologies are positioned. And of course, I mean, these are all very interesting developments, but you can see that the expected availability in this year is actually quite low. So while these are interesting developments from the point of view of technology, in practical sense, they cannot be relied on for, let's say, population scale testing, even just in a single country. Another point that's important is that for the technologies which are labeled as the outsourced device production, there is an additional logistical challenge because, of course, in many cases, the outsourced production is happening in China. And at the moment, the logistics of both the production in China and also moving the product from China to Europe or the US or other parts of the world are quite complicated. So because of these issues with the, let's say, the volumes of the tests, of the Rt-Pcr tests that are available, another technology is also becoming, let's say, quite popular and quite important. And this technology refers to, so sometimes it's referred to as serological tests, sometimes it's referred to as antibody testing. And in this particular case, what happens is that, as you can see in the diagram on the right, a blood sample is taken from a person. And what the test does is that it looks for the presence of antibodies to the SARS-CoV-2 virus in the blood of the patient. And the reasoning for this type of testing is that if the person is either having an active infection or the infection already passed, these antibodies will be present in the blood. If these antibodies are not present, then it means that the person has not been infected yet. Or let's say perhaps the person is in really early stages of infection. So the plot on the left kind of summarizes the idea. So right now, what the tests typically do is they look for two different antibodies. The one that's shown in the curve in the green and is the IgM, and the one that's shown in the curve in the red is the IgG. And the IgM will appear typically earlier in the infection cycle, so perhaps roughly 1 week after the initial infection. Whereas the IgG will appear later, and typically the IgG is the one that will be maintained at some concentration after the infection has passed. But of course, again, one of the important caveats is that this particular virus is quite versatile. So some of the applicability of the general concepts, for example, what is the IgG concentration and whether it really infers immunity and whether this immunity will be long-lasting. So all of these questions are still being investigated because we simply do not have enough data. So nevertheless, these are obviously interesting. This is interesting information to collect. And one particular, so again, in terms of practical considerations, these are the tests that are available in tens of millions of units. So in principle, these can be administered to, let's say, at the city and regional and even national levels. And in principle, they're also able to detect the so-called asymptomatic individuals. So these might be the individuals that actually have been infected, but essentially have no or very weak symptoms, and therefore would not necessarily qualify for the Rt-Pcr testing. But of course, in many cases, a positive result based on the antibody testing is best confirmed by Rt-Pcr. The specific version of the antibody testing that is, let's say, most commonly deployed are the lateral flow immunoassays. So these often in the media are referred to as express or rapid or quick, and often again described as pregnancy test-like because this is exactly what they are. And so in this particular case, as the diagram at the top shows, what happens is that we have some gold nanoparticles, which are labeled with the antigen the virus. So it means that this is not the virus itself, but certain viral proteins that have been created in the laboratory. And if they have been created correctly, then any human antibodies from the blood sample that have been created by the immune system of that individual to respond to the virus, they will actually recognize these antigens on the gold particles. And as the particles are transported across the nitrocellulose membrane, they will be captured on the lines which are specific to the particular kind of antibody. So, for example, IgM and IgG in this case. And this is indeed, a fairly simple format. And of course, this is quite rapid. So typically the result is seen in 10 to 15 minutes. And it's relatively inexpensive. So somewhere between 5 and 10 euros per test in bulk volumes. One of the challenges in terms of technology has been that in order for this test to work, the antigen, so the part that is indicated by these green squares in the diagram, has to actually be of good quality. And because this is something that has to be created, so there are many companies, many laboratories trying to create the antigens. But if the antigen basically does not match very well what the virus actually presents to the immune system, then these tests do not work very well. And so in some cases, the poor quality ones have sensitivities as low as 20%. So they really do not detect the antibodies very well. So at the moment, this is one of the challenges, and many of the tests that, for example, European countries have purchased in large volumes actually turned out not to be working well. So obviously, a lot of validation is required before we make large acquisitions. But over time, again, we expect that during this year, both the antigens and the protocols will be optimized, and so these tests will become very useful, particularly for the population level testing. As I mentioned already, in the lateral flow case, actually there is a little bit of nanotechnology involved. So there are the gold nanoparticles. But in this case, it's, let's say, fairly trivial use of nanotechnology because even, again, the regular pregnancy tests actually use the gold nanoparticles. So there's nothing, let's say, very special about it. So let's see what are the sort of more advanced uses of nanotechnology in terms of the diagnostics. So when we think about nanotechnology promise, this really is related to the general area of using nanotechnology in biosensing. That is the detection and sometimes identification of biological, usually molecules, using some type of nanotechnology. And the rationale for doing so is that definitely a lot of the biology that we're trying to detect, let's say, is itself at the nanoscale. So whether we're talking about the viruses, which are sort of roughly 100 nanometers in diameter, their components are definitely sort of smaller than that, but again, kind of on a nanoscale. But also individual biomolecules, proteins, the antibodies, the antigens, DNA. So all of these things themselves are basically nanoscale objects. And it's effectively makes sense that if we're trying to work with nanoscale objects, then perhaps using some tools that are at the same scale can be beneficial. The other motivation for using nanotechnology is that often it can bring increased sensitivity. And this comes from the fact that, for example, as indicated by this kind of, let's say, artist conception that you see on the right of some DNA molecules on a gold surface. So typically what we will try to do is we will try to bring the objects that we're trying to detect in proximity to a surface, or perhaps to some nanomaterials, such as nanowires or nanoparticles, or perhaps some nanostructured materials. And because of the special properties of the nanomaterials, we can typically then take advantage of some nano-specific chemical, electronic, optical, magnetic, and other properties to actually detect the presence of these molecules. So, for example, in this in this image on the right, you see how, for example, a DNA probe that is attached to a surface, whether a flat one or a nanoparticle, can be used to capture a target from solution. For example, in this case, you can see one forming the double-stranded configuration. And so, in principle, by taking advantage of this type of sensitivity, that's sort of one of the areas where there has been quite a lot of effort in recent decades. And so, this is definitely a promising area. Another one is obviously using nanomaterials as labels. So, good simple examples are things like quantum dots, if we're trying to do optical detection, magnetic nanoparticles, if we're trying to do magnetic detection. Gold nanoparticles are useful for many different purposes. And again, it's sort of interesting because some of these technologies are useful. For example, magnetic nanoparticles are a useful technology because coming from the IT industry, the same technology that's used to read the magnetic hard drives actually can be used to read the magnetic nanoparticles. One of the challenges is that in all of these cases, again, what we're looking at are chemical, electronic, optical, magnetic, and let's say properties which are related to the presence of biomolecules, but not actually necessarily specific to them. And so often the specificity for these types of detection schemes have to be done using biorecognition elements. So again, in the image on the right, we're using DNA as a biorecognition element. And that actually tends to be the one that is, let's say, a little bit easier to implement in practice. And if one is trying to show sensitivity limits, often DNA assays are the first thing that that people try to do. In case of using antibodies, it can be a bit more challenging because they're subject to some of the, let's say, biorecognition complications related to nonspecific and fouling and denaturing and some of the other challenges. So often after people have demonstrated in a simple sample, high levels of sensitivity, actually quite a bit of time has to be spent trying to optimize the detection scheme for, let's say, more realistic samples, more complex samples. Another sort of word of caution as far as the optimism about the sort of rapid incorporation of nanotechnology into the practical diagnostics is the fact that, again, so most of the time, the focus on the increased sensitivity by the people who develop, let's say, the new technologies for biological applications. So they often consider that essentially the limit of detection, so being able to detect increasingly decreasing concentrations or copy numbers of biological objects or molecules is the most important part. And of course, I mean, it is an important aspect, but we have to recognize, so as the diagram on the right tries to illustrate, that in general, when we're trying to do diagnostics, there are many other parameters that are also important. So there might be limits of quantification. So not just being able to detect that something is present, but in fact to try to quantify it. There are things like linear and dynamic ranges, because again, different analytes may be present in very different concentrations. There are things like resolution. So if we can detect a certain concentration, but what is the concentration? What are the two concentrations that we can reliably, let's say, distinguish from one another? And what the diagram on the right is trying to illustrate is that one of the, let's say, the reason why it's important to remember all of these parameters is that they are not completely independent. So typically, if we're trying to adjust one of them, we're also going to be adjusting some of the others. And so it is basically not really possible to, let's say, enhance all of them. So there is always some type of a trade-off, which has to be specific to the particular, let's say, diagnostic purpose. And again, as I mentioned in the previous slide, so the challenge is that the real patient samples are quite complex. So often we have to use some type of technology to pre-treat the samples to basically make them a little bit easier to analyze. And of course, again, we have to spend quite a bit of time optimizing the protocols and the formats of how exactly the new kind of detection schemes can be used. Nevertheless, nanotechnology is definitely going to be quite important in terms of the response for this specific pandemic, partly because this particular virus is really a complex target. So it exhibits different behaviors interacting with different hosts. And also, there are already kind of early indications that it looks like the that the host immune system plays an important role in determining how the disease, whether it happens and how it progresses. So effectively, it really means that what will be really important in coming months and years is the ability to not just look at the virus and not just look, for example, at the antibodies, but look at many other biomarkers. So some of the signal molecules, things like RNA, perhaps white blood cells, and many, many other, basically as many as possible, different biomarkers and many different sample types. And trying to take this information, try to correlate it with the disease progression, and to basically try to make better predictions and better understanding of both what the virus is doing and how the patients are doing. And of course, for these types of multimodal diagnostics, it will be really important to bring new methods because many of the traditional ones are not, again, scalable for large numbers of people. And of course, nanotechnology will bring both new materials and the new methods. And this is one of the big motivations to speed up the research and development in those areas. All right, so to finish my presentation, I will mention a couple of efforts at INL that are specific to the response to the pandemic. And in a most general sense, INL has basically come up with a plan that includes several generations of technologies, realizing that we have to take into account the maturity and the development lead times of the technologies in order for them to become available as soon as practically possible. And in this plan, basically the generation 0, let's say, technology is actually would be the commercial lateral flow tests that I mentioned a few slides ago. But in this particular case, as I mentioned, it's very important to validate and, in fact, go through the approval process of the commercial lateral flow tests that are available. And INL is doing something already about this at this point. And of course, the other issue is that if we're trying to deploy these tests in thousands and potentially millions, it becomes very important to provide software and hardware to assist in the readout. So on the right, the top diagram, for example, shows that typically it's a visual readout, so some lines appear. But the challenge is that, for example, if one thinks about a testing site where we need to test, somewhere between 100 and 1,000 people per hour, if we try to read, let's say, and introduce the data for these 3 lines manually, that actually very quickly becomes impractical. So we need some kind of technology which will take, just a few seconds to read these lines so that they can be uploaded to databases and so on and so on. And so these are the things that we're focusing on in the short term. So this is kind of weeks to a couple of months time scale where potentially these things can be deployed. For the first generation, the idea would be to essentially improve the existing both PCR and lateral flow technology. So to make them simpler, faster, in particular, to make them point of care. So something that can be deployed, broadly, something that can be used in not necessarily just clinical settings, again, at the different field testing sites and so on, give the answers as quickly as possible. And again, so this is maybe the time frame of perhaps, let's say, 6, 9 to 12 months. And then we're talking about the second generation. And so this is the multimodal diagnostics that I mentioned in the previous slide. And so this actually will be the situation where the existing and some of the new technologies will have to be packaged for mass production. So, for example, in the format of disposable cartridges that will be used with kind of analyzer machines. And so again, in this case, the technology advances would have to be adapted for being packaged in such a format. And of course, again, then hopefully these multimodal diagnostics will become widely available, again, both at clinical sites and perhaps even at the field testing sites. And so this is already sort of 1 to 2 years time frame, because again, there is quite a bit of development that is still necessary for this to happen. And of course, ultimately, some of the new nanotechnologies and related technologies will be adapted for the COVID response. But basically, that is, again, the longer time frame, so perhaps 2 to 3 years, because many of the low TRL technologies will need quite a bit of development before they can be commercialized. Just to give a couple of examples from the recent round of FCT funding. So one of the projects at INL that has been funded is a project about adapting one of these new variants of PCR called LAMP that I mentioned a few slides ago. And in this particular case, basically, this would be something that would be applicable to the swab samples, but will be using very simple and inexpensive, let's say, supplies. So just a small microtube with an extraction column. And then because this is one of the single tube processes that only requires a fixed temperature, so it basically means that it's something that can run using very simple equipment, or can be adapted for, let's say, simple equipment. And in this case, it can be as simple as just the detection by eye. So, for example, looking for a color change. So this is one of the ideas where basically an existing technology can be, let's say, improved, enhanced for the faster deployment. And another project is an example of a more advanced, let's say, application of nanotechnology. So in this particular case, we're talking about the surface-enhanced Raman spectroscopy, which requires nanostructured substrates, such as the one shown at the bottom. And basically, in this case, the idea is that it can be adapted. So it has been already, let's say, demonstrated for detection of, for example, DNA mutations for detection of cancer. But in this particular case, a similar system can be used to detect the viral RNA signatures. And so in this case, it's one of the advantages is that in principle, this allows for quantitative detection, and it is compatible with the microfluidic chips, as is shown on the right. So this is one of the examples of the uses of the nanotechnology specific, let's say, techniques. But of course, again, for example, this would take more than just a few weeks to be ready for, let's say, serial production as a commercial device. And so just to give sort of summary conclusions and a bit of an outlook. So I hope that I managed to convince you that definitely there is already quite a bit of advanced technology that's being deployed. And again, the pace at which it has happened is basically unprecedented. There has been no other circumstances whether within just a few months so many new things have come online. Nanotechnology is already used, and in particular will become important in the future. So I've shown you just a brief outline of the plan that INL has in terms of the several generations of the diagnostic technologies. And of course, while we already have many partners, organizations, companies, and so on, but one of the purposes of this presentation is to make sure that those of you who have recognized some areas where you think you can contribute, we definitely would like to talk to you. So please make sure to get in touch. And with this, I thank you for your attention, and I think we will proceed to the questions.
Thank you, Dmitri. We now have some time for questions. Please type them in the chat on the right corner of the screen.
All right, so the first question is about the sequencing. Well, in principle, ideally, it would be excellent if we could, in fact, sequence the virus from every patient, because already the sequence analyses are very useful for epidemiology. So to try to understand how the virus has been spreading. So, for example, some of the findings include the fact that in the United States, much of the virus appears to have been brought from Europe rather than directly from China. But at the moment, obviously, we are quite far from being able to do that. Nevertheless, we have to remember that some of the technologies that have been developed after the Human Genome Project, right? So at this point, there are some devices that can perform at least, let's say, some level of sequencing within just a few hours and at a cost of, let's say, roughly €100. So while this is definitely not going to be something that we will be able to do for millions of people, but I think that adapting some of the sequencing technologies, and of course, I suspect that both the protocols and the devices can be optimized to work with the viral genome, because again, it has its specific properties, and it's actually not as large as the human genome. So this would definitely be an important contribution. I have not actually seen very much, let's say, information, whether in terms of marketing, let's say, or other announcements about this, but this will definitely be an interesting direction. And I suspect that the companies that have been making the devices for human genomics will also look in this area. So, okay, next question. Will the investment in nanotechnology be higher after the pandemic? Well, I mean, I think I would say in general there I would be optimistic, partly again because this is one of the situations where let's say some of the promises that have been made by the advances in biotechnology and nanotechnology are already, let's say, being fulfilled. So again, right now we're benefiting from all of the advances that have happened, for example, after the previous, SARS epidemic. And again, in particular, because different types of testing and multimodal testing and all of those other things, they will be important. And again, we're talking about years, because even, for example, if we think about even when the vaccine becomes available, it will still need to be actually followed up whether the vaccination is working correctly. So even just within the pandemic context, once we're out of the crisis situation, this will be sort of relevant for years. And of course, I think now many people realize that this is this is more of an alarm, because effectively we expect that similar virus events may happen in the future. And so we basically have to have the system ready. And of course, nanotechnology is one of the ways, one of the key enabling technologies that is very important. In particular, just to kind of indicate one of the areas, right? In particular, nanotechnology offers one of the ways to create relatively inexpensive and kind of simple to use devices, which nevertheless have quite high performance characteristics. And this is definitely one of the aspects that INL, among many others, is working on. Maybe the next question. Okay. Projects on the development of active protection, being able to act against the virus. Yeah. Well, at INL we have submitted actually some of the proposals related to the use of nanotechnology and the, let's say, related types of coatings, both to try to, let's say, better sanitize and reuse some of the protective equipment, and potentially and let's say, but in case of the virus, I think one of the challenges is that because often people think about some of the antibacterial uses of nanotechnology, but virus is a bit more complicated, because effectively, in many cases, the way nanotechnology kills bacteria relies on the fact that it interrupts certain vital, pathways in these bacteria. The problem with the virus is that it's actually not alive, and therefore there are sort of fewer opportunities to try to interrupt something. Nevertheless, I mean, some of the emerging information is indicating that, for example, the survival rates of the virus on different types of surfaces are very different. So I'm confident that with time, we will discover some of the, let's say, nano modified surfaces that, for example, will sort of decrease the survival time of the virus and so on. So these are definitely areas of very active research, but also, again, because this is a virus, some of these things are actually more complicated because in some sense, it is not very alive, so it's not so easy to kill it. The use of nanoparticles like metal oxides to extract RNA from a solution containing a sample from a patient in order to avoid the use of reagents that are in short, I suspect, in short supply. Well, again, so sure, some of the, and in particular, the use of nanoparticles, it can be important, let's say, it can be important in diagnostics, partly to address the question of, that I mentioned, that in some cases, the samples that we collect may contain only a very small number of the copies of the virus. So maybe there is 1,000 or 10,000 in something like 1 milliliter. So often having some kind of a platform that is using nanoparticles can be beneficial because effectively the nanoparticles can be mixed with the sample, let's say, homogeneously, and therefore they will have a chance to capture at least some, both encounter and then capture at least some of these targets. So in the specific question about the RNA, actually this is perhaps not the first area that I would think about, simply because the RNA amplification methods don't actually require the RNA to be, let's say, completely removed and separated from the other parts of the solution. So they work, as I mentioned, so for example, these LAMP methods and so on, it's just a single tube. So you more or less place all the reagents there and the process goes. It is true that some of the reagents, have to be produced, but this is actually one of the areas where, let's say, the community that is working on synthesizing the reagents is catching up fairly quickly. So I anticipate that specifically the shortage of reagents for nucleic acid assays will not be, let's say, a long-term problem. There are actually some of the more interesting situations where once we start thinking about the antibodies against the virus, again, not the human antibodies, but the antibodies against the virus. So there will be some interesting developments because, again, there actually the traditional methods of producing antibodies may not be, let's say, fast enough. Well, any insights on immunosuppressed patients? Well, I think at the moment, right, let's say one of the biggest concerns, which is not specific to immunosuppressed patients, but related to immunosuppression, is actually the fact that in the most critical cases, so the people who are likely to die from COVID infection, in many cases, what actually kills them is the so-called cytokine storm. So it is basically the overreaction of the immune system that essentially leads to the to this outcome, not the virus itself. And so in this particular case, but it's actually a very difficult situation because on one hand, the doctors can try to apply, in that case, intentionally immunosuppression. But of course, again, if you suppress the immune system too much, then the virus will kill the patient. So this becomes, an extremely difficult thing to balance. And again, this is one of the hottest, let's say, areas of research where the doctors are trying to find some combinations of the treatments. But of course, in case of the patients that are immunosuppressed, whether naturally or using the drugs because of some of the other issues, this, of course, is going to be a big challenge. So this is one of the areas where some of the methods, like the antibody therapies, right, where if we do find that the patients who have passed the disease, at least some of them develop effective antibodies, particularly the blocking type of antibodies. So basically the antibodies that prevent infection by blocking the functional parts of the virus. So, for example, being able to extract those antibodies and then introduce them to the patients whose own immune system is not able to fight. This is one of the promising channels, but of course, again, that's a very difficult channel because it more or less requires large numbers of human donors and so on, and processing of those antibody samples. But this is also one of the active areas of research, and I believe in Portugal as well there are some efforts on this. The impregnation of masks and textiles with a virus resistance nanocoating could be a good solution, and an active protection able to kill the virus given by nanocoating could be viable. Well, this is a little bit related to one of the earlier questions. So again, in this particular case, the challenge is that the concept of killing the virus is different from the concept of killing a bacterium. And in many cases, when we think about antibacterial coating, what those coatings tend to do is they basically, again, they tend to disrupt, whether chemically or sometimes mechanically, some of the vital pathway inside a bacterium, and basically just so they really kill them. In case of the virus, what effectively has to happen is that we have to promote essentially the damage or disassembly, for example, to the viral particle. And to some extent, this is one of the reasons, right, why the health authorities are saying that we should use soap when we wash our hands, because in fact, soap is quite effective at disassembling the viral particles. So in that sense, the types of coatings that could be really effective against the viral particles are likely going to be different from the coatings that, for example, that people have been developing for bacterial resistance. But nevertheless, so we do know, even though there are very few, I think there's only a couple of studies so far about the virus survival on different surfaces, but we do know that the type of surface does make a difference. Unfortunately, for example, for the time being, the surface that has been established to be the least friendly for the virus is basically copper. But of course, copper, again, is toxic to many living things, including ourselves in large amounts. But in principle, yes. So there is evidence that surface treatments it should be possible to find ones that will help to deactivate the virus. But it will probably take a few months before we actually find the efficient ones and the ones that can be deployed at large scale. Okay, do you consider that the new devices and testing methods can compete with thermal cyclers in terms of cost? Right. Well, this is the cost is an interesting issue, and I partly sort of I only mentioned it a little bit during the presentation, but of course, in terms of practical deployment, let's say this is important. So my colleagues who are working in this area are telling me that, for example, for the Rt-Pcr, basically just the cost in terms of the reagents and labor per test is roughly 50 euros if it's done in Europe. And then respectively, typically sort of the retail level, let's say, cost is in the range of 100 to 150 euros per test. So in principle, using the current technology, the costs are already substantial. One of the areas, let's say, where some of the new devices and technologies are going to be competitive is in the point of care situations, because obviously in the centralized locations where they may have some of these systems that can run 100 samples in parallel. And again, so they basically have the conditions, they have the staff to prepare the samples and so on. So such systems can be very efficient. But of course, when it's a centralized location and such setups tend to be, the large ones tend to be quite expensive. So typically there may be, just one or two sites in the country. And therefore, samples have to be carried there. And in some cases, simply moving the samples will take several days. And then right now, one of the suspicions why often the Rt-Pcr tests are failing is because something goes wrong with the samples in transport. So in many cases, the new devices and technologies are trying to go for something simpler, where the device itself will be smaller and less costly. So maybe less than 10,000 euros, maybe even less than 1,000 euros, and such that they can be really positioned in many, let's say, local areas. And therefore, they would be able to basically process the samples as they're being collected to avoid, the delays and other logistical challenges in moving them to central locations. So these technologies, they are really all complementary because they, in terms of cost, in terms of performance, and other sort of situations, they have, let's say, complementary features. So I don't think that one or the other will, let's say, defeat, or let's say there is something to be won. There is definitely room for all of them. And again, in fact, as a slide that I showed indicated, for the moment, even all of them combined still do not provide sufficient number of tests this year. So I think that in this case, it's not going to be so much the competition, but really trying to deploy as many of them as possible. However, obviously maintaining the quality of the testing, because poor quality tests are very problematic at this point.
Thank you, Dmitri. Unfortunately, we don't have time for more questions. Thank you for your presence, and see you soon at Fibrenamics Forum.
Thank you for your attention, and thank you for the very interesting questions.